Hello everyone, welcome back to my YouTube channel. This channel is called Life Sciences by Tumi. This is a channel where we share life sciences content to make sure that you ace your exams, you ace all your formal assessments. If you are enjoying the content in my channel, please do like, subscribe, and leave anything in the comments, any questions that you might have, any contribution that you might have, and please do share the link to this video as it will. My name is Tumi Silani. I am a teacher at Milner High School here in the Northwest Province. This is a lesson series on the DNA Code of Life. Before this lesson, we had 2 more lessons prior to this video. Be sure to check those out. This is lesson 3 in our lesson series of the DNA Code of Life. It is titled DNA Replication. Before we get into the lesson, let us firstly look at the lesson breakdown of the series. This series is broken down into 7 lessons. Each lesson contains its own video. So like I said, so far, we did lesson 1, which was on the revision of the structure of the cell, revising content that is relevant for grade 12 only. So we looked at the organelles, the structures, the functions, in terms of grade 12 content. So be sure to check that video. On the previous lesson, we looked at the DNA structure. On lesson 3, which is today's lesson, in this particular video, we will be looking at DNA replication. However, for you to understand the DNA replication content, it is important for you to understand the DNA structure. So please pause or leave this video and go back to the DNA structure, where we discuss the structure of DNA and how it functions, for you to be able to quite understand the content on this particular lesson. On the following lesson, on a separate video of course, we will be looking at the history of the discovery of DNA. So we will be studying a quite controversial story on how DNA was discovered. On a separate video, we will have lesson 5, which is on the other nucleic acid that is called RNA. We will be looking at its function. Lesson 6 is a 2-part lesson on protein synthesis. It is divided into the process of protein synthesis, which comprises of transcription, movement of mRNA, as well as translation. The second part on the lesson of protein synthesis will be looking at amino acid coding. Lesson 7 is the last lesson of the series. We will be looking at DNA profiling. We will be describing, or rather defining, DNA profiling and looking at its uses. We will be looking at things that compromise the reliability of DNA profiling, and we'll be doing some investigative work when we interpret the different DNA profiles in order to determine paternity, and we will be looking at different DNA profiles in order to determine a possible criminal in a forensic or criminal case. Okay, I hope you're ready. Let's just move directly into today's lesson. Before we look at the process of DNA replication, there are a few introductory concepts that we need to look at, and then we'll move into the process of DNA replication. All right. As for the introduction of DNA replication, the first thing that we need to do is define what DNA replication is. What is it that we are talking about when we talk about DNA replication? So simply, DNA replication is the process where DNA makes identical copies of itself. Please use the word identical here. Don't use the word the same or similar or whatever. It is the process where DNA makes identical copies of itself. So the copies of DNA should be identical, 100% the same. From the previous lesson, we learned that DNA has specific codes for our different characteristics. Those codes are represented by the sequence of nitrogenous bases that appear on the DNA strands. Therefore, if DNA makes more of itself and it is not identical, that results in a mutation. You will learn about mutations specifically in term 2 on the topic of genetics. Yes, in the topic of genetics. So, DNA replication, the DNA must make identical copies of itself, or else a wrong code will be made, a mutation may occur, and that may lead to detrimental results. All right. So put simply, DNA replication is the process where DNA makes identical copies of itself. That definition is worth 2 marks. The second question, where does DNA replication occur? Okay. Now, in lesson 1, in that video, we said that DNA is found in 3 places: the nucleus, the mitochondria, and in plant cells. It is also found in the chloroplasts, if you can still remember. Now, most of our DNA is found in the nucleus. Therefore, here we will be describing DNA replication as it occurs in the nucleus. Okay, so the answer to this question would be DNA replication takes place in the nucleus of a cell. The third question, when in the cell's life cycle does DNA replication take place? Now, the cell, just like us human beings, have its own life cycle. It has stages that it passes through until it dies. All right? Now, this question is asking of when. It is asking for a phase in its life cycle where DNA replication takes place. And the answer to that is it takes place during interphase. All right? So the word interphase refers to the phase or the period in time in which DNA replication takes place. Now, let us look into what interphase is. Okay? You need not know a detailed description of interphase in grade 12. You learned about interphase in grade 10. In grade 12, on this lesson, I am going to share information that is only relevant for grade 12 purposes. I would not be looking at interphase in a greater detail. So, what is interphase? Interphase is a phase. The word phase means period in time. Okay, guys? The word phase is a period in time. So we are describing a time where something happens. So interphase is a phase or a period in time before cell division. So this is before mitosis or before meiosis, where the cell prepares to divide. Okay, by mitosis or by meiosis. So when you talk about interphase, we are not talking about a process. We are talking about a period in time before cell division where the cell prepares to divide. So interphase is not part of mitosis. It is not part of meiosis. It is a phase or a period of time before cell division. And what happens is that the cell is preparing to divide. Okay? Next question. What are some of the occurrences that take place during interphase? So what happens during interphase? DNA replication takes place. So if you get a question that says what happens during interphase, I doubt that you're going to receive that question. But if, let's say, you get that question, you are going to say DNA replication takes place. Look at question number 3. Question number 3 is asking when in the cell's life cycle does DNA replication take place? Okay, and we said it takes place during interphase. So during interphase, DNA replication takes place. The cell doubles its DNA or its genetic material. The cell doubles its DNA or it doubles its genetic material. Why? Okay, before we go to the why, unreplicated chromosomes, guys. Still remember this from lesson 1. Unreplicated chromosomes will become replicated chromosomes. So as the cell prepares to divide, DNA replication takes place. And the cell's genetic material or the cell's DNA is doubled. Remember, a chromosome is essentially DNA that is wrapped around histone proteins. So unreplicated chromosomes will become replicated. If you can still remember, what are unreplicated chromosomes? These are chromosomes with only one chromatid. Okay? Now, these chromosomes contain DNA. So if these chromosomes contain DNA and they have one chromatid, when we double that DNA on that one chromatid, we will be creating another chromatid. Therefore, unreplicated chromosomes that have one chromatid will become replicated chromosomes, and replicated chromosomes are those chromosomes that contain two chromatids. Okay, now we will look into that in the following slide. I will make a more detailed explanation of this concept on the next slide. Okay, here's a note. Watch the video on lesson 1 for the full breakdown of what a chromosome is. Please do go back to that lesson after this lesson to look at what do we mean by an unreplicated chromosome versus a replicated chromosome. On this slide, I will be going back to the content that I did on the video of lesson 1 into explaining what is a chromosome. This is to bring a little bit of context to the question that we looked at in the previous slide. Remember, you might get a question that asks you to explain the significance or the importance of DNA replication. This question is requiring you to explain the effect of DNA replication in terms of the distribution of the genetic material or the distribution of DNA into the daughter cells after cell division. Okay, guys? Now, the question might be, explain the significance of DNA replication or explain the importance of DNA replication. And the answer should be, during interphase, DNA will make identical copies of itself, 1 mark, thereby doubling the cell's genetic material or doubling the cell's DNA, 1 mark. Unreplicated chromosomes will become replicated chromosomes, 1 mark. And after cell division, the daughter cells will receive all the required genetic material from the mother cell, or the daughter cells will receive all the required DNA from the mother cell. That is the final mark. So this question carries a total of 4 marks when it is fully explained. Now, For us to understand the difference between a replicated chromosome and an unreplicated chromosome, let us firstly look at what a chromosome is. Okay? Remember, I do not own any rights to the videos that I include in my presentations. Hence, I've attached the sources from which the simulations, from which I got the simulations in my presentations. So to explain this, I will be using a video simulation. Let us play it and check what is happening. So there we have a cell, okay? When we move into the cell, where did we say we find most of the cell's DNA? We find most of the cell's DNA inside the nucleus. Now we are going to move inside the nucleus through the nucleopores. And then when we look inside the nucleus, we said we are going to find DNA. Now from lesson 1, we said that when the cell is not dividing, DNA will be arranged or packaged into threads that are called the chromatin network. However, when the cell is dividing, this DNA, these threads of chromatin will start to compress, and then we will start seeing, or DNA will be packaged into structures that are called chromosomes, as we can see over there. However, what is a chromosome? So we see a chromosome drawn like that. We see chromosomes drawn like that mostly. It has chromatids. If a chromosome contains only one chromatid, we say that chromosome is an unreplicated chromosome. However, if a chromosome has two chromatids that are joined together by a centromere, remember we said that is a replicated chromosome. So whether a chromosome has one chromatid or two chromatids, it is still a chromosome. Now, let us continue. What do we expect to see when we unbundle, or when we break down, or when we unwrap chromatids of a chromosome? A chromosome is essentially made up of two things, okay? When we unbundle or unwrap this chromosome, we expect to see two things. We expect to see DNA and histone proteins, okay? So the two components of a chromosome are DNA and histone proteins. Now, DNA coils itself or wraps itself around histone proteins over and over and over and over, and wraps itself again and again and again and again and again to form the chromatids of a chromosome. So when we unwrap a chromosome, we expect to see DNA and histone proteins. In terms of DNA replication, if we have a chromosome with one chromatid, essentially a chromosome is a packaging of DNA when the cell is dividing. A chromosome is nothing but DNA that is wrapped around histone proteins. So a chromosome is how DNA is organized. Okay, guys? Now, in terms of DNA replication, if you have a chromosome with only one chromatid, within the chromatid, remember, we said we find DNA. So when DNA makes identical copies of itself, remember, DNA is making more of itself. Therefore, when a chromosome contains one chromatid and DNA makes a copy of that, we are going to, instead of finding one chromatid, we are going to find two chromatids. And when a chromosome has one chromatid, we said it is an unreplicated chromosome. And then during DNA replication, the DNA within this one chromatid doubles itself. We are going to find two chromatids. Still on the context of the effect of DNA replication, where DNA makes identical copies of itself, and then the cell doubles its genetic material or doubles its DNA, and unreplicated chromosomes now will become replicated chromosomes. We are now on this slide checking what an unreplicated chromosome is versus what a replicated chromosome is. Now, in your publications like study guides and textbooks and the likes, we often see chromosomes drawn like this, having two chromatids that are joined together by a centromere. This is what we call a replicated chromosome because it contains two chromatids. As you can see over there, we have chromatid 1 on the left and we have chromatid 2 on the right, and these two chromatids are joined together by a structure that is called the centromere. This is a replicated chromosome because it contains two chromatids. However, in the cell, chromosomes don't always look like this, do not always have two chromatids, all right? Sometimes chromosomes look like that. Chromosomes have only one chromatid. Okay, guys? Now, when a chromosome has only one chromatid, we say that chromosome is called an unreplicated chromosome. Okay? So, if a chromosome contains only one chromatid, it is still a chromosome, but it is a chromosome that is unreplicated. All right. Now, how does this chromosome move from being unreplicated to being replicated? The process of DNA replication has to occur. From the previous slide, we have explained that this chromatid contains DNA that is wrapped around histone proteins over and over and over and over and over and over again. Okay, now we said that a chromatid is essentially DNA that is wrapped around histone proteins. Okay, now remember what DNA replication is. DNA replication, we said, is the process where the DNA makes identical copies of itself. When we make a copy of the DNA that we find in this chromatid, what do we expect to find? So when we make a copy of the DNA found in this chromatid, a copy of that would actually form a separate chromatid. Okay, meaning a replicated chromosome would contain the original chromatid, that one over there, and then the additional or the second chromatid would actually be a copy chromatid. All right, so before DNA replication, a chromosome would only have one chromatid. All right, when DNA replication occurs, the DNA within this one chromatid is doubled. We are making more. Therefore, it will have two chromatids. Let me show you what I mean. Okay, now we said to move from having an unreplicated chromosome with one chromatid to having a replicated chromosome with two chromatids, DNA replication has to occur. Now, this process takes place during interphase. Remember, guys, please do not make a mistake here. DNA replication is the process, and it takes place during interphase. Remember, we said phase means the period in time. So the process where the process is DNA replication, and the phase or the period in time is called interphase. So this one chromatid, we expect to make a copy of itself and have now two chromatids. This would be the original chromatid, and this will be the copy chromatid. Okay. Now, chromosomes do not always have two chromatids. They only contain two chromatids after DNA replication. Okay, so as you can see over there, unreplicated chromosomes are visible within the cell before DNA replication, and replicated chromosomes are visible in the cell after DNA replication. Now, Look at it like this. This makes sense. A replicated chromosome is visible after DNA replication. So after DNA replication, the chromosome will be replicated. Before DNA replication, the chromosome will be unreplicated. Now, what does it take for an unreplicated chromosome to become replicated? We need DNA replication. All right? Okay, we are continuing with DNA replication, the introduction. Please note, you do not have to know the details about interphase in grade 12. Knowing the above mentioned points is sufficient. It is enough to know the above mentioned points. I'd like to emphasize again, interphase is a phase or period in time where DNA replication takes place, and DNA replication is the process itself. So from interphase, guys, you need to know what interphase is, what happens during interphase in terms of the bullets in the previous slides, and what is the effect of DNA replication. The following question asks, why does DNA replication occur? So here we need to describe the importance of DNA replication. Importance, it might be phrased in a question that you must provide the significance of DNA replication for 3 marks. Okay, let us explain. During interphase, DNA makes identical copies of itself. 1 tick. Thereby doubling the cell's DNA or doubling the cell's genetic material. Remember, when you make a copy, you are doubling, you are duplicating, you are making a +1. Why do we need to double the cell's DNA? After cell division, each daughter cell receives all the required genetic material from the mother cell. Remember, with cell division, what is happening during cell division? You will find a situation where you have, for example, in mitosis, you have 1. Okay? You have the original cell. After cell division, after mitosis, we need to have 2 cells that we call daughter cells. Okay? So, during mitosis, this one cell will divide into 2 daughter cells. Now remember, in mitosis, the daughter cells must be identical to each other and identical to the original cell that we call the mother cell. All right. Now, I'm just going to make an example explaining this point, that each daughter cell receives all the required genetic material from the mother cell. We said that chromosomes may be packaged in structures that are called chromosome. DNA is packaged in structures called chromosomes, if you can still remember from the previous lesson. The human body cells contain 46 chromosomes. These 46 chromosomes contain all the genetic material or DNA of a human being. All the genes, all the DNA codes. So all the DNA is contained within these 46 chromosomes. Now, cell division is dividing the cell in half to form 2 cells that must be identical. Now think about it. If a cell is dividing, a cell that contains 46 chromosomes, if it divides, we are going to have a situation where we have 23 chromosomes there, and we have 23 chromosomes there. First question of mine is that, do these cells, daughter cells, are they identical to the mother cell? No, they're not. They would contain half the number of chromosomes. And if they are body cells or somatic cells, they need to be identical to the mother cell, meaning they need to have 46 chromosomes as well. Okay? Now, this would be invalid. This would be totally invalid. All right? Now, what DNA replication does, the importance of DNA replication, I will show you just now. During mitosis, like we said, we would start off with a cell. We would start off with a cell that contains 46 chromosomes. Remember, the daughter cells must be identical to the mother cell. Okay? The daughter cells must also contain 46 chromosomes, and the other cell also must contain 46 chromosomes. Is it possible for us to divide this cell and produce 46? If we have 46 chromosomes, that's impossible. So what DNA replication does, it doubles the cell's genetic material, meaning times 2. So 46 times 2 would equal how many? 46 times 2 would equal 92. I hope my mathematics is correct. So before the cell divides, the 46 chromosomes will be multiplied by 2, meaning we have twice as many chromosomes. We have twice as much DNA from the cell. Now, when we divide 92 into 2 daughter cells, each daughter cell will receive what? Let's go back there. All the required genetic material from the mother cell, because these daughter cells must contain 46 chromosomes. So before the cell divides, the genetic material must be doubled. Why? Because the daughter cells would receive all the required genetic material from the cell. In this case, the daughter cells will receive 46. So that's why DNA replication is important. To go back, during interphase, DNA makes identical copies of itself, thereby doubling the cell's DNA or doubling the cell's genetic material. So 46 times 2 is 92. So the genetic material will be doubled. Now, after cell division, each daughter cell receives all the required genetic material from the mother cell. So each daughter cell will now contain 46 chromosomes that are carrying all the required genetic material or all the required DNA that is needed by that particular cell. I hope this makes sense, because we are now moving on to the next slide. Okay, now we have come to the process of DNA replication. What I am going to advise you here is that we are going to learn the steps of DNA replication in a specific sequence. I have divided it into step 1, step 2, step 3, as well as step 4. We are going to learn it theoretically and learn it visually as well. I don't know if the advice that I'm going to give you is good advice or it's bad advice. What I advise you to do here, firstly, is to memorize the steps, line by line, word by word, step by step. Okay? These steps also appear on the examination guidelines for Life Sciences Grade 12. So you are required to write it sentence by sentence, word by word, So there are certain concepts that you need to understand. There are certain concepts that you need to memorize. There are certain steps that you need to do, certain content that you need to do both, memorize and understand. So I'm going to advise you here to memorize. I don't want to use the word cram because cram is a bad word, but I am advising you to cram the steps word by word, letter by letter, please, word by word, sentence by sentence, the way that it is, because you will be required to write it down exactly as it appears here. Okay, now, when DNA makes identical copies of itself, how does it do it? It does this in the following steps. We know that originally DNA is a double helix, meaning it has two strands and those two strands are twisted. So when DNA is like that, when DNA is a double helix like that, it cannot replicate itself. Okay? So what needs to happen first before it starts to, what should happen first? So that, like I said, DNA, when it is like this, it cannot really replicate, okay? Cannot make copies of itself. So step number one, the very first step of DNA replication, let me just erase there. The very first step of DNA replication is that the DNA double helix unwinds, okay? Now, when it unwinds, it will become straight, such as there. DNA double helix unwinds. There it is. Step number two is that the weak hydrogen bonds break. Now, remember, DNA has two strands. The left strand is there, the right strand is there. These two strands are joined together by weak hydrogen bonds in the middle over there. Okay, so step number two, the weak hydrogen bonds break. Check what is happening to the weak hydrogen bonds. The weak hydrogen bonds are breaking, as you can see. Remember that the weak hydrogen bonds are the ones that are holding the two strands together. Now, after the weak hydrogen bonds break, what is going to happen? Step three, the two strands of DNA will separate, or the two strands of DNA will unzip. So you only need to write one. You can either say the two strands of DNA separate or the two strands of DNA unzip. Let's check what happens to the strands. There we go. The strands have separated. The thing that was keeping the strands together is now gone. The weak hydrogen bonds, they have broken. So when the weak hydrogen bonds break, the two strands of DNA will separate. Both strands act as a template. Now, let us describe this word. What do we mean by a template? Think of a template like a foundation on which you are going to build something. For example, before a house is built into a huge structure, the people that built the house will firstly lay a foundation down. So a foundation of a house is a template on where we are going to build a house. We are going to lay bricks on top of that foundation to build a house. Another example, think of a slice of bread, for example. A slice of bread is not a sandwich. It is a template. It is a foundation. It is a template where we are going to add butter. We add cheese, lettuce, whatever. Now, when we are adding these other elements, we are building a sandwich. So when you talk about a template, we are talking about a foundation on where we are going to build another strand of DNA in order to make a DNA molecule. Okay? So a template is a foundation where we are going to add things. And what are we going to be adding? You will see later what we are going to be adding to these templates to make two identical DNA molecules. So going back, DNA double helix unwinds, so it essentially becomes straight. The things that are holding the two strands together are called weak hydrogen bonds. So the weak hydrogen bonds will break, and then once the weak hydrogen bonds are no more there, the two strands, what will happen to them? They will separate because the two hydrogen bonds are no longer there to keep them together. Why do these hydrogen bonds need to be weak? Because they need to be easily broken by enzymes during DNA replication. Hence, we call them weak hydrogen bonds. If they were strong, they will not be able to be broken by enzymes, and therefore, if they are unbreakable, DNA will not have the ability to make copies of itself. After they separate, both strands will act as a template. Okay. Let us pause there and let's check what is happening here on this video simulation. I want you to take a moment and check what is happening. I will rewind this video and explain to you exactly what is happening there. Okay, let me just pause. What do we see there? This thing that we see over there, let me just go back. Okay, now, what do we see there? We see a double helix, DNA in its helical form. Okay, so the first step, we said the DNA double helix, the part where it is replicating will unwind, okay, and become straight. And the weak hydrogen bonds between the two strands will break. This does not happen magically. The weak hydrogen bonds don't just magically break. These weak hydrogen bonds are broken by an enzyme. So this structure that you see over there, or that molecule that you see over there, it is an enzyme that is breaking the weak hydrogen bonds between the two strands of DNA. So when it breaks the weak hydrogen bonds between the two strands of DNA, the two strands of DNA, as you can see over there and over there, those strands will start to separate. Okay, you need not know the specific names. It is not in our grade 12 curriculum to know the specific names of the enzymes. But all you need to know that every single step, every single occurrence, every single thing that is happening during DNA replication does not just magically happen. Every single step is controlled by enzymes. So this is the enzyme. I'm not going to name the enzyme to confuse you, because you don't need to know the names of the enzymes. Okay, so this enzyme breaks the weak hydrogen bonds. When the weak hydrogen bonds break, you must check the two strands of DNA will separate. Okay, let me continue playing the simulation. So as the enzyme is breaking the weak hydrogen bonds, please do observe what happens to the DNA strands. The DNA strands, like I said, separate, and both of them form what we call a template. All right. Okay, continuing. Step number 4. Free-floating DNA nucleotides. Free-floating DNA nucleotides from the nucleoplasm. Remember, this process is happening, like we said in the previous slides, this process is happening in the nucleus. And in the nucleus, all the material inside the nucleus is floating in the nucleoplasm. The nucleoplasm, remember, we said is the watery substance, the fluid that we find inside the nucleus. Okay, so free-floating DNA nucleotides. Guys, please do not just say free-floating nucleotides. You must say free-floating DNA nucleotides from the nucleoplasm attached to both templates in a complementary way. Remember, the complementary base pairing in DNA, we said that, if you can still remember the analogy, the apple always falls from the T. So adenine is always complementary to thymine, and the car always parks in the garage. So cytosine is always complementary to guanine. Apple falls from the tree, meaning A goes with T, and then the car parks in the garage. So C goes with G. So in the nucleoplasm, we have free-floating DNA nucleotides. They're just floating around the nucleoplasm. Now, it is like they are drawn to this template, to these two templates. Immediately when the two strands separate, we said they become templates. Now, as they become templates like this, it is the perfect opportunity for these free-floating DNA nucleotides to attach themselves to the template in a complementary way. So it is important to write in a complementary way. If you don't want to write in a complementary way, you will say free-floating DNA nucleotides from the nucleoplasm attached to both templates. And then you will say A will bond with T, and then C will bond with G. Now let's check what happens. Look at what's happening. The free-floating nitrogenous base, the free-floating DNA nucleotides, it is the whole nucleotide that is traveling, guys, not just the nitrogenous base. Okay, it is the whole nucleotide that is traveling. Remember, a nucleotide is made up of a phosphate group. It would have a sugar portion, and it will also have a nitrogenous base. So it is the nitrogenous base that is complementary. Okay, however, a nitrogenous base does not only travel alone, it travels with the rest of the nucleotide, meaning it travels with the phosphate group and the sugar portion as well. So what are we doing here? We are checking this step. Free-floating DNA nucleotides, which are those from the nucleoplasm, attached to both templates in a complementary way. Okay, so look at what is happening. Free-floating nitrogenous bases are attaching themselves to the template in a complementary way. What do we mean in a complementary way? A will bond with T, or will be complementary to T. G will be complementary to C. That is complementary base pairing in DNA. Now, remember, both strands act as a template. Now, this is the original strand, which is a template. When the free-floating nitrogenous bases, when the free-floating DNA nucleotides, one by one, attach themselves to the template in a complementary way, we are building another strand, a strand that is a copy. Now, we are using the original strand as a template to build a complementary strand, okay, in a complementary way. Now, let us check on this strand what is going to happen. Essentially, the same thing is going to happen that happened on this strand. The free-floating DNA nucleotides are going to attach themselves to the template in a complementary way, meaning when we see A, it will be complemented by T. Where we see C, it will be complemented by G. Now, we have an A in this template. Which nucleotide carrying which nitrogenous base must flow and attach itself there? It must be a DNA nucleotide that contains nitrogenous base what? Thymine. Apple, tree. The next one will contain adenine. Following one, guanine. The next one, cytosine. Following one, guanine. Following one, adenine. Next one, thymine. Following one, just like that. So they are attaching themselves in a complementary way. Remember, guys, I'm going to repeat this again. The original strand that separated became a template. Okay, so that template attracts free-floating DNA nucleotides to it, to attach to it in a complementary way. Now, the original strand is the template. As these free-floating DNA nucleotides attach themselves there, they are creating another strand of DNA, as you can see over there. Okay, let us check this video simulation. Nope, let's just skip it. There's another one that I'll show you later on. Now, lastly, step number 5. So after the free-floating DNA nucleotides have attached themselves to the templates in a complementary way, 2 new DNA molecules are formed. 2 new DNA what? Not 2 new DNA strands, you guys. 2 new DNA molecules. Now, at the marking center, at the end of the year, when we mark your scripts, usually we see learners make the mistake of saying 2 new DNA strands are formed. Nope, not 2 new DNA strands, 2 new DNA molecules are formed, each with 1 original strand and 1 new strand. Let us check. There we go. So we have formed 2 new DNA molecules. 2 new DNA molecules. Each molecule contains an original strand that was a template and a new strand. This one also contains the original strand, and that one is a new strand, as you can see over there. Remember, DNA replication, the DNA makes copies, identical copies of itself. When we check the DNA code in this piece of DNA, it must be the same as the DNA code in this piece of DNA. AT, AT, TA, TA, CG, CG, GC, GC, CG, CG, TA, TA, AT, AT, TA, TA, GC, GC, AT, AT, CG, CG, TA, TA, and GC, as well as GC. So the 2 DNA molecules are identical to each other. So there is no mutation here. However, if there's a certain mistake that happens, for example, this T is replaced by a G. This one over here is replaced by a G. We call that a mutation. And like I said, we'll talk about mutations at a later stage. More visuals showing the process of DNA replication. Okay, let's look at more visuals. Now, please, like I said, go and memorize, go and cram all of those steps of DNA replication the way that they are. And now when you visually see the steps of DNA replication, you will be able to attach what you have crammed, what you have memorized to the individual steps. So step number 1, we said that the DNA double helix unwinds. There we go. It is straight. Step number 2, we said that the weak hydrogen bonds between the 2 strands break. And then when that happens, the 2 DNA strands are going to separate. All right. So here we had weak hydrogen bonds. When they break, then the 2 strands are going to separate. And we said that when they are separate like this, both of the strands act as templates. Step number 3 What did we say? We said that free-floating DNA nucleotides will attach themselves to both strands in a complementary way. Now, there are the free-floating DNA nucleotides. They are being attached to the templates in a complementary way. Now, when they attach themselves in a complementary way to the strand, these DNA nucleotides are building another strand of DNA. Therefore, at the end, two DNA molecules will be formed, and they will contain an original strand as well as a new strand. The last visual that is showing the steps of DNA replication. Let me just play it for you. Check what is happening. This key over there represents an enzyme, right? So this enzyme, its job is to break the weak hydrogen bonds, okay, between the two strands. Let's check. Look at what the key is doing. It is breaking the weak hydrogen bonds. Let me go back. Look at what the key is doing. It is breaking the weak hydrogen bonds. Now, when the enzymes break the weak hydrogen bonds, what did we say is going to happen? We said that the strands are going to separate. Two strands are going to separate. There they are. They have separated. And then we said that the following step, both of them will act as a template. A template for what? A template for free-floating DNA nucleotides to attach to. Okay? For free-floating DNA nucleotides to attach to. And once the free-floating DNA nucleotides attach themselves to both strands, two new DNA molecules, two identical DNA molecules will be formed, one with an old strand and one with a new strand. So, guys, go back and revise, or rather go back and memorize those steps of DNA replication so that you can associate the content to the visuals. If you did not cram those steps, if you did not understand those steps, I can play this visual the whole day. You will never understand what is happening there. All right. So please pause the video. Take 3 to 5 minutes to try and memorize those steps. Maybe write them down, have them on the side. As you are checking the visual, you check what's happening step by step. Let me continue the video. So, we said both strands act as a template. So as they are acting as a template, look at what's happening there. Here is a free-floating DNA nucleotide. It is coming from the nucleoplasm to attach itself to the template in a complementary way. There we go. At the end, what are we going to have? There are our free-floating DNA nucleotides attaching themselves to the templates in a complementary way. Okay, now what I want you to observe, there is the template, right? And at the top there also, that strand acts as a template. Now, what's going to happen is that the free-floating DNA nucleotides are going to attach themselves in a complementary way. So that the free-floating DNA nucleotide that attaches itself to that template over there needs to have a complementary nitrogenous base. So there it must be a nucleotide that has a nitrogenous base A, their T, their T, their A, and their A as well. Was I right? Absolutely, I was. Now, at the end of DNA replication, we are going to have a situation where, let me just go back there. At the end of DNA replication, we will now be having two DNA molecules that, and when you look at them, when you look at the codes in terms of the sequence of nitrogenous bases, they are identical. Okay, guys, I hope this made sense. Okay, now we have reached the end of lesson 3. Unfortunately for this lesson, I do not have any assessment questions for you now. I've planned the assessment questions for DNA replication after. However, your homework is to go back and memorize the steps of the steps of DNA replication, the way that they are, and associate each step to the visuals that I've given you in this video lesson. Okay, if you've enjoyed this lesson, please give me a like. If there's something that you do not understand, put that on the comments. I will read all your comments and make sure that I respond to as many as I can. Subscribe so that my channel does not disappear on your YouTube. And to benefit other learners as well, please do share the link to this video. Thank you very much for watching. The next lesson will be based on the history of the discovery of DNA. Thank you and goodbye.